Welcome. In this video tutorial, I will briefly describe the output possibilities Advanced Cell Classifier provides, and I will show you how to create reports of your data. To obtain output from Advanced Cell Classifier, the user must have previously defined classes of interest with annotated cells and must have a trained classifier. To have an overview of what Advanced Cell Classifier can provide as output, please open the classification menu and read the labels of the items. The main output format is a cell-by-cell classification, incidence and distribution of the different classes for each plate, and class-based statistics for the different features computed. Let us start with the button Predict Current Image. It allows the user to check the result of the automatic classification directly by looking at each image. Practically, it shows the class automatically assigned by overlaying a number in a colored rectangle for each cell. Make sure that the cell number button is on, otherwise you cannot see the results. If the user is not satisfied with the classification obtained, he or she can manually correct the improperly classified cells and then train the classifier again to check the new results. Once satisfied with the cell-by-cell classification, the user can generate reports for the full plate using the Predict Selected Plates button. A setting window will appear. At the top of the window, you can simply select the plates which will be included in the analysis. Below this, two lists allow for the selection of the classes of interest. All of them can be selected, and the classes to be used as normalization factor to compute the well-by-well incidence ratio, technically called hit rate. The hit rate is computed by dividing the number of cells belonging to the selected classes by the number of cells classified in one of the normalization classes. For example, to see the well-by-well ratio of rounded up cells, select Rounded Up on the left and all the classes in the right and press Start. The program will save the report files directly to the metadata folder, typically named Anal2, of the analyzed plate. Let's see where the results are located. By navigating into the Selected Plates metadata folder, we can see that several report files have been generated. Let's take a look into the first one, platename.csv, which is a comma-separated file. Every row in this file represents a well of the current plate. In each row, there is the name of the plate, the indices of the well, the total number of cells, the hit rate, and finally the number of cells in each class. In this example, in the B4 well of plate test 1, there were 78 cells in the standard class and 95 cells in the rounded up class. Class names are indicated in the header of this file. Advanced Cell Classifier also provides the user a way to automatically compute mean, standard deviation, median, and median absolute deviation separately, considering for each well the cells that belong to the different classes. To obtain the report file containing the different feature-based statistics for the different classes of cells, simply click on the Feature-Based Statistics button. The file will be automatically saved in the metadata folder. Now you have gained insight into Advanced Cell Classifier's report generation methods. To learn more about the different output files and parameters available, please read the help documentation file provided with the software. Thank you for your attention.